let me just say that I'm not a physicist or a mathematician, so it's kind of unusual for me to present at a physics and mathematics type of conference. I'm a neuroscientist, medically trained, so mostly focusing on consciousness from a medical and human and biological standpoint, right? I'm going to talk about a new phenomenon that we discovered, which may have something to do with consciousness. I'm going to try to convince you that that is the case by actual experimental data. And but I want to tell you a little bit about how I got into that act. As a as a motivation for me to do the experiments that I'm going to tell you about, and that involves some physics, but not involving equations or anything. Whatever is known in the popular physics literature, pretty much. So you would have to suspend disbelief for at least the first part of my talk, because it will involve some really wild ideas about new physical principles that by chance turn out to be confirmed by the experimental findings that I've got. But I'm not sure if those physical interpretations are real. So I would encourage people, physicists among you, to come up with a physics explanation for some of the data. So I'm not going to make any claims about the theoretical aspects of my presentation. Okay, I just want you to concentrate on the data, and that data is solid, so it's reproducible, it's robust, and you cannot fake it. And I'm going to show you why you cannot. I mean, we've done experiments to show that you cannot fake. We are not fooling ourselves, in other words, right? So from a scientific standpoint, let's not talk about philosophical, implications like the hard problem and other things. So I'm just concentrating on very practical questions and some scientific questions. So these are questions that every person who is interested in consciousness would like to know the answer to, right? So what's the physical nature of consciousness? Is it just electrical activity of the brain or information processing within the brain, or some other explanation that is already known, or phenomena that are already known about the brain? So if we can answer that question, then what's the neural physical code of qualia, elementary elements of consciousness? And then, of course, two practical questions. Can we measure the level of consciousness with an instrument? Can we tell whether an organism is conscious or not using that instrument, right? And then final question is important from an evolution. If you believe that consciousness emerges out of the nervous system somehow as an evolutionary process, then it would have to have adaptive advantage, right? So what is that advantage? Does the physical mechanism of consciousness that we discovered, let's say, have causal significance for brain function? So if it does, then obviously that would have an adaptive advantage for evolving as you go down the evolutionary ladder. So those are the questions. So just to tell you that I'm at the end of my talk, I believe, and I want you to see if I can, I want to know if I've convinced you of that. I believe we have answered question number 3 and question number 4 to our satisfaction, at least. And the question is whether you are satisfied, and I would need feedback on that issue. And of course, there is some hint of what the physical nature of consciousness is from what we find, and also what the physical mechanism, the adaptive advantage or causal relationship of the physical mechanism to the brain function is. And those are the 4 questions that we have kind of addressed from what I'm going to tell you about. So the thing that started all this for me was taking seriously this notion that consciousness is a serial process. This has been popularized by William James at the turn of the last century. in his big classic book for the father of psychology, William James. That's the William James I'm talking about. And then, of course, a lot of other writers and philosophers, as well as psychologists, have popularized this notion from actual data, which is that each conscious moment has limited information holding capacity as it transitions sequentially from one moment to the next. It's like one damn thing after another, is what you know, in a colloquial term, and that's what consciousness is. Therefore, one can, from, again, now getting into sort of the physics mathematics of it, which I'm not really competent to deal with from a analytical standpoint, but this is what I started out with. Therefore, one can model the underlying brain activity that codes for conscious experiences as a time series of unfolding activity patterns. You know, and so I started out with that. And about more than 20 years ago, I published a paper, purely theoretical, again, not involved with any equations or anything, but purely conceptual extrapolation of that idea, which was that conscious experiences are generated and coded by small, non-arbitrary, universal activity, subset of spatially localized temporal neural activity patterns. So the activity patterns that code for conscious elements of conscious experiences need to be temporal in nature because it's a time series, right? Then the patterns are, again, this is coming from that same, it's a small subset. And what defines that subset? They may be mathematically related to each other somehow and distinct from all other patterns that are non-conscious, okay? And the patterns are then repeated multiple times over short sub-second periods because of network oscillations, which we know from neurophysiology exist within the brain. And the intensity of those associated experiences will then be a function of how many times you repeat those patterns. Then repetitions have the function of potentiating synapses, right? So this is Hab's postulate, those neurons that fire together, wire together concept. So, and that is the repetitive firing thing. And then once you have that notion, you can have limitless variation of these temporal pattern, different combinations. You know, you can concatenate that. You can have different temporal sequences of those patterns in different forms. So going from that, again, another sort of influence on me was these two quotes, one by John Archibald Wheeler, who's kind of the father, one of the fathers of quantum mechanics, that no phenomenon is real until it is an observed phenomenon. And that is, you know, he said it in a different context, but at the end of my talk, you will realize that it applies to what we found. Okay, and then of course, Sir Roger Penrose, it may well be that there is something else going on in the brain that we don't have an inkling of at the moment. And that's, again, also at the end of my talk, you might see an inkling of what it might be. And so that's what I'm going to tell you about. Okay, so here's my leaps of faith, not coming from physics, but purely speculation. So I often tell people when I give this kind of a talk that because I'm not a physicist, I'm not constrained by the rules of physics, right? So, I can speculate wildly. So the two leaps of faith I made was, what if the coding, so instead of, when I proposed the hypothesis that I showed you about more than 20 years ago, it was electrical pattern, neuronal activity, electrical activity patterns. So what I imagine is, what if the coding temporal patterns are not electrical activity patterns, but they're a pattern of increasing and decreasing, increases and decreases of wave function collapses in time. Okay, so everything is the same, except it's now collapsing wave functions. Of course, these are spontaneous collapses. So that, again, is a whole kind of upheaval of existing physics. And Sir Roger talked about that, and the collapse of the wave function is the critical element. The second leap of faith that I made is, what if this pattern at a given focus within the brain or epicenter within the brain can spread in an expanding volume of space in the body and outside the body by inducing collapses of wave functions in the surrounding region, replicating the original source pattern. Okay, so those were the two kind of assumptions. So if you do that, you obviously have a different, you have a quantum universal temporal pattern hypothesis, which everything is the same except the wave, it's not electrical activity, but there's sequences of wave function collapses in space and time. And so if you talk about the source or the focus or the epicenter of such an event, it would spread out in space into the brain, into the body, and outside into the space surrounding the brain and the body. And so that's where this insight came, for me at least, is what if we have a device that measures these collapses of wave function? And the classical, classical not in the class, a quantum classical device is a double-slit device, right? So where if it's a wave, you get an interference pattern. If it's a particle, it's a photon, you get those two bands. So you have a dichotomy that you can measure. Okay, how do you measure it? Okay, you can make a device. So it's right here. Okay, this is the device. You have a double slit. I'll also tell you about something else, but basic thing is you have a laser diode that shines laser through double slits, and then it samples, there are, in this case, 5 photodiodes, photosensitive diodes, that sample the interference patterns falling on them, right? So if collapse occurs right at the beginning when it emerges, you would have a tendency to produce 2 bands. If collapse doesn't occur and it's a wave, then you would have it spread out as an interference pattern. So if you sample the intensity of light, in the peripheral photodiodes, not the central one, just the peripheral one, you will see intensity variation. So if collapse was induced by whatever that happens in the brain, then you would see less intensity. If collapse didn't occur, then you would see the same level of intensity that you would gather. So it's a pretty simple thing. And so you can get this little sensor unit containing that, and you can bring it close to your head, because I'm talking about spreading in space as well, this effect, right? And so this is basically, I'll show you this again over in the picture. Okay, so there's another way we discovered later on that you can do it much simpler. Rather than having a double slit device, all you need is a laser diode that shines light, and then you're sampling the periphery of it, which is basically diffracted patterns or beam divergence patterns, which have diffraction is also a wave phenomenon. So decrease in diffraction, those diodes that are there in the penumbra of the laser focus that you're shining, those diodes, they will sample intensity. If it's a collapse and a photon, then you would have less intensity there. If it's a wave, you would have greater intensity in the penumbra. So it's basically the same thing. In fact, this one is this device rather than the double slit apparatus, because it's a much more elementary, much more minimal device to measure what I'm going to show you about. So this is the device I'm telling you about. So basically, it has this sensor unit, which is this. Then you have a controller and an A to D converter. So it takes the analog signal, converts it into digital, and you attach it to an iPad or a Android tablet or a laptop. You measure, it's a serial monitor that samples from four of those diodes, right? Photodiodes, and then plots them. That's the idea. It's a pretty simple thing. So if you do that, right? So you have, this is for the interference device. You have inner bands and outer bands. Okay, if you put this device in the middle of your lab and you move out, right? There's nobody in the lab. There's no consciousness in the surrounding medium. You just have this device. What you get is a flat recording pretty much. So from all 4 channels, right? So intensity kind of flat. And then why kind of transform it for, so you would like to know if anything that happens to any of those channels, whether it's common to all 4 channels, okay? That variance that you produce, is it common across those 4 channels? And so I do simple principal component analysis, and it turns out that the first principal component covers more than 97% of the variance across all 4 channels. So that gives you an idea that if it's not just a random fluctuation independently affecting each channel. So you have something that's common, right? And then for a better display, I'm an electrophysiologist by training, so I have to do EEG recordings, intracellular recordings from neurons and all that. So we used various ways of making little data look beautiful from a visual standpoint. So one approach would be do a running average of it, right? Every few, in this case, every second. So these are sampled at 100 hertz. So every 10 milliseconds, you take a reading. You can, our device can go up to, I think, about 1 kilohertz, definitely 500 hertz, right? So, and then I do a final normalized display, which just involves subtracting the first time point, the amplitude at the first time point from all other time points. And that sort of inverts the response. And you'll see why that is important, but I mean, from a visual standpoint. Okay, so I showed you when nobody was there, right? So now if you bring your head close to the device, to this sensor unit, like this, all of a sudden you see a nice transient that you can see. 10 minutes exposure produces a nice pattern that almost looks like what I used to measure before from single neurons, what is called excitatory postsynaptic potential. This is from a whole human being, right? So you see a nice increase and decrease, and it actually, the time course is over tens of minutes, so 40 minutes pretty much, right? So, and so the next slide. And it turns out that the deep that you see in the raw channels that you sample is 34%. So there's a 34% decrease in intensity in the 4 channels that you're recording from, and which translates, when you invert, we do my little analysis, it translates to 810 arbitrary units with this device. I call them centurions. Just to, I think, and the device we have labeled is a sentiometer because we think it measures sentience, another word for consciousness in the colloquial language. So, okay, so you might ask, is this some artifact that we are picking up, right? So we did lots of standard, ruled out all the standard possibilities. It's not because of body heat, because 37 degrees bath, which is our body temperature, doesn't produce much of a difference. Produces a slight response, which may be due to change in the sensitivity of the electronics of the device, but certainly not as much as you see with the human head. Ultra-weak photons, biophotons, which is kind of in fashion today, are not responsible because if you turn off the laser, you don't see it. Respiration, sounds, and waves of pressure, air pressure, are not responsible because if you put the device in a vacuum chamber and you bring your head close to the wall of the vacuum, you still see a response, and I'll show you that actually. Radio frequency waves are not interfering with this recording. Response is not blocked by Faraday bag, if you put the device in a Faraday bag, and we have also done it in a Faraday chamber now. Magnetic field response is not blocked by magnetic shields, shielding material, new metal or whatever. It's not electric field coming out of your brain because response is not completely blocked by aluminum, any of the metals we tried, right? So here's in the vacuum. So there's no difference in whether it's vacuum or air. Here's with a copper shielding. There's no difference. I mean, no reduction for sure. Okay, so then are we fooling ourselves, right? So classic thing in all of medical clinical literature is double-blind studies. So here's we had two devices, two chairs far apart from each other. One chair had an active device, the other chair had inactive device, and only the active one was recording. And then the subject was in a room, researchers stayed outside. So, and the subject did musical chairs. They went from one chair to the other chair at random intervals. And then we were recording the whole time from one of the active devices. And so whenever he sat on the actual active chair, we saw this response. Whenever he sat on the other chair, we didn't see much of a response, right? So it's double blind. So neither the researcher nor the subject knew where the device was. Then these are low-hanging fruit experiments. That's what I call them. So if you have a device, and remember I said decline. I'm close on time, so. Okay, sorry, yeah. I've got a lot of stuff to do. I mean, I'll just zip through it. So, pardon me. So, yeah, it's okay. So, I told you that the hypothesis, my loose hypothesis was it would fall off with distance, the response. And that is what we find. And then it's roughly 1 over r square, 1 over distance square. So it's an inverse square law finding, okay? If you change the position of the device with respect to your head, you see a difference. The forehead shows less. The two temples show larger response, left being bigger than the right. Orientation of the device itself is important. The orientation of beam relative to the head is important. So if the beam is horizontal and moving away from your head, you see a bigger response. You see the biggest response. With the other orientations, you don't see much of a big, I mean, you still see a response, but not as big. Okay, so now how is it related to consciousness? From a medical standpoint, I'm in the Department of Psychiatry and Neurology, so we do sleep studies, right? So easy thing, sleep is a consciousness phenomenon. So you put the device near a sleeping subject, what you see are those oscillations, right? So you see rhythmic oscillations. They don't correspond to the sleep periods, the cyclical periods they are actually shorter than the sleep cycles. Okay, but perhaps they're dreaming cycles, right? The person is dreaming or not dreaming, deep sleep dreaming, deep sleep dreaming. It's known now that dream is not just a RAM phenomenon, rapid eye movement phenomenon. It's also, dreams happen also during non-REM sleep. So the cyclical aspects of dreaming might be much faster than what you record with an EEG, which is used by all sleep labs everywhere, right? So, okay, the other low-hanging fruit is, what about different organisms, right? We started with animals. So vertebrates, human, of course, mouse shows the same kind of thing. Then we went to crayfish. Crayfish, we had 5 crayfish, right? These are crustaceans. Then, they move around in the field in shallow water. Out of 5 fish, 3 of them, crayfish, we got a positive response. One of them, the remaining 2 got a negative response. I kind of initially dismissed that negative response. What is inverted consciousness? If this is consciousness that we are recording, what does the opposite of consciousness mean? It's kind of weird. So I thought, oh, this may be something going on there. But then I brought a crab, a blue crab, a whole bunch of blue crabs, and recorded from them. They all showed inverted responses. Then I brought cherry stone clam, right? So these are pretty fat clams. And then recorded a whole bunch of cherry stone clams, and I see them all inverted, right? And then finally, so I had sampled crustaceans, I've sampled mollusks. Now the next thing that remained in the invertebrate, the big other family was annelids, which are worms basically. So we got California black worms. These are really tiny worms, like 1 to 2 inch long. So one worm might not have an effect on this thing. So we let them, they reproduce by asexual reproduction. They split into 2. So, and binary fission. So we got a whole bunch of them, made a whole bunch of them, let them reproduce. And so I think there were like 500 of them in a little vial of water. And then when we did just a 15-minute exposure, which is what we did for others, we didn't see much of a change. There was a slight inversion. But when we did a 60-minute recording, you see huge change. These are tiny. I mean, they don't virtually have nothing in terms of a nervous system. We're very, very tiny, right? So then, of course, plants, right? Other low-hanging fruit. So plants for 15 minutes, I saw kind of a tendency for an inversion, but some of the plants were just flat, right? But then we did 60 minutes, and you see a big inverted response in plants. What the heck is inverted consciousness, right? Well, opposite of consciousness, I don't know. Okay, but it gets predicted from the basic idea that I had, where I said it's increases as well as decreases in collapse of the wave function. So if you had more increases compared to decreases, then you would see a positive response. If you have more decreases compared to increases. Sorry, so then I will, the next thing about consciousness is anesthesia. What happens if you anesthetize? We did that in a mouse, right? You find that the anesthetized amplitude of the response goes down, as you would predict, because anesthesia makes you unconscious. And the other thing which I had not expected is that the rise time decreases as well. So it becomes a faster rise and a lower amplitude that's shown there, right? So, and it turns out that it's the rise time that is critical for lower consciousness, because we had mice, especially with ketamine xylazine anesthesia, that when they recovered, their amplitude didn't recover, their rise time only recovered, right? So, yes. And okay, I'm going to, so this, yeah.
Yeah, it's just as a drinks reception upstairs, like a briefing. I mean, I think we could go to some questions.
Can I just do a summary slide? So I'll just zip through the next three.
Want to go, and the rest of us will. Oh, that would be polite, perhaps. Perhaps we can kind of find something in the middle.
I'll just zip through 5 slides quickly, okay, and then I'm done, okay. I'll apologize for what I'm going to show you, because I'm killing mice and they're under humane treatment, So what happens after death is the issue, right? So, okay, so we kill mice. So we find that amplitude goes down, rise time goes down, and and that remains for 65 minutes. And after about 2 hours, it flattens out, right? So next one also I have to apologize for. What if you separate the brain from the body, right? By doing a decapitation after killing the mouse using humane methods. So what you find is interesting. So basically the body acts like a passive vessel, because this brain is spreading the response throughout the body. So body shows, the torso shows just a decaying response over time. But the brain modulates that response. Initially you see a biphasic response in the first 65 minutes. And then after about 3 hours, you see an inverted response, as if we become, or the mouse became an invertebrate. It's not, it moved from being a vertebrate to after death. So there is something going on. Obviously, it's not electrical activity, because electrical activity shuts down within a few minutes of killing the mouse. Yeah, here's another one. So the final thing is basically what happens to, what is the baseline due to, right? And it turns out, so we did a long recording of 18 hours. You see that when researchers were leaving the lab, you saw a decrease. They were far away. They were not close to the device. It's like, so this is long distance effects of the consciousnesses of people working in the lab. And then when they came back to the lab in the morning, you saw an increase. Okay, so basically what I'm saying is, let me go to this. So I'm saying is, we have dissociated consciousness from the brain electromagnetic activity. And so it is, it outlasts the death of an organism. So whatever process, this quantum wave, if it is a quantum process involving wave function collapses, it occurs in a molecular network within the brain. independent of energy, a bioenergetically independent network, and possibly a microtubular type network or a mitochondrial network or whatever, but it's molecules within the brain by themselves going off on their own. And then there is a bidirectional influence between the brain electromagnetic activity and this activity. This activity influences the brain activity and then vice versa. And that explains our general anesthesia and clinical death effects. I predict that psychedelic state would show a partial severing of the bidirectional connection so that the molecular network goes off on its own to some extent, mildly controlled by the electromagnetic. Anyway, this is what we are making next, what we call qualiography, where we are trying to actually decipher, mine the patterns, temporal patterns from the responses from the brain. And these are the people who helped me with the whole project itself. And thank you very much. Sorry for the